Okay, so I wanted to thank Neelima, Ram, and Amit for organizing this. I wanted to start with a little introduction of our lab and where we are. But before that, I wanted to encourage you to approach us. We are here because of you guys. We really want to get to know you, and we are open to all sorts of questions you have. We also in the program have discussion sessions and all that, but I think for you, it's going to be even much better if you come and talk to us. We are super friendly. We like to talk to you. We want to meet you. And that's the whole point of making this huge trip from Argentina to here. So please do that. So our lab is here in South America, in Buenos Aires. And so we travel all the way like up to here. So, and it's a kind of jet lag. So we'll see. So we are at the University of Buenos Aires in the north part of the city of Buenos Aires. Actually, Neelima was there in December, and also Ram was there, I don't know when, 5 years ago, yeah, 19. Okay, and this is our department is in the campus. So we have this building that it's called Zero and Infinite, because that's how so it's a new building. We are super happy with it. And in campus, so University of Buenos Aires is a huge university, hundreds of thousands of students. It's the most important university in Argentina. And I wanted to tell you a little bit about our lab, that it's in the physics department of Exactas, is the School of Science, how we call the School of Science. And I wanted to give you an overview also because of Gabo's talks, so that you have an idea of all the things that we do. So in our lab that it's called Dynamical System Lab, that is like a very open. There inside the lab you can do whatever you want. So that's why it has a non-specific name. So the goal of our research is to shed light on the dynamical mechanisms involving the generation of complex sounds and to characterize the function of the peripheral system and the central nervous system in this process. So our approach is that the brain and the body interact in order to generate behavior. So we are always in the problems try to look at not the detail of the neurons or the specific, I don't know, neural ion channels, but it's more like a wider perspective. And considering that all this is immersed in the environment, and that can affect all the dynamics. Okay. Okay, something collapsed. It's coming? I don't know. It's kind of, maybe I didn't.
Yeah, this button.
Thank you. So in our lab, we do physics and neuroscience of vocal production. So we integrate physiological recordings. So we are in the physics department, but we have birds in our lab. We have zebra finches and canaries. And here we can record respiratory activity or muscle activity while the bird is singing. So in that way, we are recording the physiological instruction that the bird is doing while it's generating the behavior. While that is happening, we can also do neural recordings. So I am in charge of this line in our lab. So we can do surgeries, neural surgeries, in order to insert electrodes and record neural activity while the bird is singing. And I will show you a little bit in my talk. And also while the bird is sleeping or it's anesthetized, and we can do several experiments here. We also do field work. So this is, so we've done field work like 20 years ago. But it's a line of work that Gabo started during the COVID pandemic, where we couldn't go to our lab. So Gabo say, well, we have an immense lab that is the wild, the nature. So let's go to nature. It's full of birds. Let's do some experiments there. And so there we work with chingolos that Gabo will tell you about, and horneros. Also, it's a local species. Both are local species, but these kind of species are species that are only in South America, so that's an advantage. And we integrate all these experiments with modeling. So that's the key point. Not only doing different kind of experiments, but also integrated in modeling. So we can do modeling about neural activity or the avian apparatus, so physics of phonation that apply to songbirds. And also in the last years, integrating this with tools of artificial intelligence. Okay, so this is like a broad view of our lab. Also, our work has been in the news a lot, not because we are doing, birds is not that they are not interested in the physics community. Physics Today and the IIISER has commented our work. Also, this was commented with Scientific American because we also do some lines of how to interpret birdsong dreaming, and Gabo will tell you about in his talks. And we are super happy with this last work that was also commented in National Geographic, and we are super happy. But I won't get deeper in this because Gabo will tell you about in his talks. So why we study songbirds? How we look songbirds? Well, we have 10,000 species of birds. Among them, around 40% learn to sing. So the ones that learn to sing and songbirds, for example, canaries, parrots, nightingales, so mostly all the birds that sing pretty, mostly they learn how to sing, parrots and hummingbirds. And why is this interesting? It's because, I don't know if this is working, so. Because it's not so often in the animal kingdom that vocal learning occurs. And also because these learning stages are very similar to human learning stages. And what is vocal learning is to modify the sounds that you're producing through imitation. For example, monkeys don't learn how to speak, so to vocalize. They can learn how to set meaning to these vocalizations, but the vocalizations are innate. So it's hardwired in what they, so it's what they have. They grow, and it's all what they have, these vocalizations. So this ability of changing the vocalizations, copying a tutor, is very, very weird. Other species that has vocal learnings are whales or dolphins, all these kind of species, elephants, some bats, and that's it, and humans and birds. But also, so they are great animal models to study vocal learning, but also to study motor control. Because if you think about it, you have to control many muscles and the respiratory activity and all that in order to generate the vocalizations. And I tell you, think about what you're doing, because the physical mechanisms of song production in birds are very similar to the human mechanisms of speech production. So depending on what you're interested in vocal learning or motor control, you can use bird songs as songbirds as animal models, okay? Okay. So after this, so I don't know, well, I'm losing track of time, but let's see. I'm okay.
Anna, I just have a question.
Yes.
Because in some of these animals, I don't know. What about dogs? Is that an example of learning or?
Yeah, no. So dogs. So that is. So dogs or cats and all that. They learn. So they learn how to code what you're. So they can learn stuff, but they cannot modify the vocalization. So they have one vocalization that it's coded in their genes, and it's fixed there. So it's not that they can modify it in order to copy something. That is the general knowledge. So they can learn a lot. They can learn how to memorize what you are saying. So for example, grab me that or their name or do, so they can learn a lot of things, but not so, so the current knowledge is that they need specific genes in order to modify the vocalizations that are not present.
More familiar instance because we see a lot of dogs and we don't see a lot of whales.
Dogs it's weird because I mean it happens with my dog. Then I think it looks like she's kind of vocalizing in a specific time when I arrive home. She vocalizes different when she's trying to, I don't know. It's fighting with the neighbor's dog. But that is something that it's innate. It seems like, okay? So that's a current knowledge. It could be that with more research, we discover that they have some flexibility, but the main picture right now is that they have something that it's innate. It can be that they have, for example, it could be that some dogs are special because they have something that is different from other dogs. So the Einstein of dogs, for example. And that can happen, but all that is coded in the genes. So it's not something that you can learn through experience. That's there. Yeah. Another question? Something, yes?
About fish. What about
fish vocalization?
Usually they communicate. I mean, I don't know enough about it, but
so they can communicate. So so that's the thing. So and not only fish, so many animals. So they can communicate through vocalizations, and each species has its own vocalization. So vocalizations in fishes, they are also it's not so well studied, but there are some studies there, and they can communicate in the same size, for example, as monkeys or whatever. So other species that say, well, you had different vocalizations and assign those vocalizations to meanings. And that is also that that is learned through experience. So you teach your offsprings, well, this vocalization is because I wanted to show you that there is some danger. And this vocalization is because there is food there. And they learn to pair meaning to vocalizations, but it's not that they can change the vocalizations in order to generate new knowledge. That would be the idea. That's the idea. Yeah. Okay. So let's move on to my talk. So this was like an introduction, and I wanted to tell you a little bit about rhythmic neural oscillations underlying birdsong. Okay. So Ran, tell me what time should I finish?
You should try to finish at 11 so that we have some time for questions.
Great. Okay. So to start my talk, I wanted to tell you that there's also some rhythm in the speech. So this is just to highlight the similarity between human and songbirds. And so this is the sound of a person speaking. This is the sonogram, that it's a representation of frequency and time. And this is the envelope of this sound. And you can see that here, if you look at where the silents are, there is some rhythm. Well, some student that did her PhD in our lab studied this in her postdoc and find that it's some speech rhythm there. And also that this rhythm is correlated, they can record from the brain, from specific regions in the brain that are dedicated to speech processing. And they see that these oscillations in the brain correlate with the oscillations in the speech. And also that synchronizing to speech enhances perception. So there is some rhythms in the human brain, and it's related with behavior. So the question is, well, if you look at the brain of a bird, this is the brain of a bird, and this is the brain of a human, and this is in the same scale. So they can be very different. So the question would be, could it be the case that you find similar rhythms in a somewhere brain? Well, the idea is that it can be very different, but if you look at regions and functional regions in the brain, you can find some similarities, so there is still hope. So we wanted to study rhythms in birds, so I wanted to show you a little bit about the behavior before going to the neurons. So again, so this is a canary song. Let me see if I can. Would you hear that? Okay. I don't know. But in any case, if you couldn't hear, it doesn't matter. So here is the representation, so time and the frequency. For example, if you were hearing like beep, 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 is this that you have like a fixed tone there. And we can also record the air sac pressure while the bird is singing, as I told you before. So these are expiratory pulses. So, and during the expiration is when they generate the sound. And so each of these expiratory pulses corresponds to a note here that we call syllables. And the first thing that we did was characterize these syllables. Are all these, so acoustically they can be different, but if you look at the motor gesture that we call, so the representation, this can be better if I just, were you listening? Yeah, maybe this is better. Okay, don't touch it, okay. So our question was, so for example here, these are two different syllables because acoustically are different, but if you look at the pressure pattern, they are all like sinusoidal signals. So we characterize that and we say, well, you have actually, what it looks like is that you have 4 types. So we did a PCA analysis, or you can do other type of analysis. Now with machine learning, you can do several type of that. But it looks like you have 4 groups. And these 4 groups are characterized with these specific shapes of the pressure pattern. So you have 4, and each one of these groups has a specific rhythm, and these rhythms are classified by motor gestures. So you have 4 groups of syllable rates. Okay, so you can have different acoustics, but we are not being like focusing in the acoustic, but in more like this, like in what are the rhythms, what are the specific shapes of that behavior. So with this in mind, we're recording neural activity. So these are the rhythms that we want to see if they are coded some way in the brain. So for that, we do these neural recordings. So this is the raw data that we extract when we put an electrode inside the brain. And you can do 2 things. You can do a low-pass filter from this signal and you get the local field potential, so these slow oscillations, or you can do a high-pass filter and you extract the spikes that are the way the neurons communicate. And so with this, for example, so the neurons fire at a specific syllable. And what we can do with these spikes is look at them isolated, or you can set a threshold and count spikes. And with that, you can get like a profile of activity. So this profile is just doing counting spike and putting a sliding window and just counting this spike, and then you smooth it a little bit. But the idea is whenever you see a peak here, it's because you have many neurons firing at the same time. So peak is a lot of neurons, not something that is flat. It's because you have not so many neurons, okay? With this in mind, one experiment that we did was playing back the song, what the bird is singing. We call it boss because it's birdsong song. So each bird has its specific song that was learned through a sophisticated process. And yes.
It's a response to the bird's own song. You take a recording of the bird's song and you play it back to the bird?
Exactly. We record the bird first and then we have a recording and we play back to the bird. This we can do while the bird is freely behaving, while they are anesthetized. So these recordings are through anesthesia, or when they are sleeping. You have like all these three behavior states, but this is like that. So this is the bird is anesthetized. We play back the bird's own song, and we see how the neurons respond to that. Exactly. And so with that, so we use this electrode. So this is the representation. You have like 32 channels. And in each channel, I am showing you here the repetition of 20 trials. So as this is biology, you have to do with a lot of trials and also with controls and all that. And this is the response of that. So, so one thing that you can see is that all of this is like synchronized. So in the different channels, you see kind of the similar responses. So you can do this with multi-unit activity, or you can do with LFP. So what I was showing you here is that you can get either multi-unit activity or local field potential. Both of them have its own oscillations, but you can see they are well synchronized. We quantify this and along all the 32 channels. And this is a representation of what you are seeing here. So there is a region where you have it's a high correlation. And when it's correlated here, it's correlated here. So this is why the same channels are in this signal. And then if you don't have a signal like here, mostly it's because you are already outside the region of interest. Okay, so as they are well correlated, what we are going to do for this part is just taking one channel of all these recordings, one channel that has the highest amplitude. And with this, again, the question was, are there rhythms? So now we have the tools. We have the behavior and we have the neural recordings. And the question was, are there rhythms in a songbird brain? And so one first result is that if you get, so this is just a part of the song, but I'm zooming so that you can see that you have each syllable. So this is the sounds. So again, so this is the sound that you are playing back to the bird. So this is a representation of the syllable, and this is our LFP recordings. So each of these lines is a recording in a specific site, getting to the channel of maximum amplitude. And you can record so you have two hemispheres of the brain, and you record in a place that it's called HBC, it doesn't matter, it's a region of interest. And you can record in the left hemisphere and in the right hemisphere, and in all of them you have all these sinusoidals that first of all, they are also, you get the same oscillations in different regions of the brain. But then if you just see, well, where are the valleys, and you drive it here, you can see that they are all synchronized with the song, okay? So when we first find this, well, here are the rhythms of the brain. So these oscillations are well correlated with the sounds, okay? So as always in science, first you have the observation and then you have to systematize all your results. And so we did this for different syllables, and in all of them, so for example, look at the time. So for example, different syllables have different durations. And for example, these are very long syllables that can last up to 200 milliseconds. And you have slow oscillations, but these oscillations are also mimicking the syllable. So whatever the syllable is a long syllable, the oscillations are long. If the syllable is short, the oscillations in the brain are matching the rhythms, okay? Another way of looking at this, before I was just showing you parts of individual syllables, here I'm showing you the whole song that I'm presenting to the bird. And you can see that here, so to me, this is also a nice striking image. And here is here is the LFP, the oscillations. And this is a sonogram of these oscillations. So whatever you see a defined line is because these oscillations have a defined frequency and they are in 16 hertz, okay? So here also you have another oscillation, and these slow oscillations, you can also see some defined thing like around 3.7 hertz, okay? So slow oscillations is because you have those syllables. Faster oscillations like 16 hertz is because you have faster syllables. And also, the frequency of the LFP correlates with the frequency of the syllable rate, okay? If you just process this and you plot the syllabic rate and the local field potential rate, you can fit this in with a line that is an identity, okay? So the syllabic rate corresponds to the local field potential rate, okay? So this is what we are saying. You have rhythms in the brain, in a songbird brain, and that rhythm corresponds to behavior. And we can discover this because we choose a specific species, that is the canary, that has their song with these specific rhythms. There are other. Can I have this? Yeah, thank you. I was not sure if I was drinking some others. So choosing the right species of birds is also an important thing for your discovery. Okay, so this was. Yeah.
Rhythm in the brain is just an electrical signal that you are measuring.
Electrical signal in the brain, yeah. And these LFPs are collective. So we are collective activity in the brain. So this is because we have several neurons that are synchronized. Exactly, and that's why you can see like all these rhythms. Yeah.
Is it one of the standard waves, the theta wave, and or is it something else?
Sorry?
The brain signal.
Yes.
Is it one of the standard waves like the theta wave, or is it something else?
Well, we started like that precisely. When we started recording LFP, we started to first look at the theta rhythms, gamma rhythms, and all these specific frequencies. But it was not so clear until we discovered, these are not theta rhythms, are syllable rhythms. That was Santiago Boari, my PhD student, that he was analyzing this. I said, well, look, if I just look at which are the rhythms of the syllable, that's where these rhythms are. If I am just looking at the previous knowledge as theta or gamma rhythm and that, it will just be that rhythm whenever that rhythm has to do with this kind of rhythms. So yeah, so it was not that, it was more like syllable rhythms. But we started looking in that direction. Yeah. Okay, so this was the result of anesthetized birds presenting the recording of the song. So the question is, well, what happened when the bird is singing, when it's generating the behavior? So I wanted to show you the data. So this is how we record in the lab. We use tetrodes. So we have 16 channels. So each of these is one electrode. The only thing is that in each row, these electrodes are very close to each other. So we have four electrodes close to each other. And this is another electrode that it's kind of far away. And this is another electrode and so on. So you will see here is the bird singing. And here we can select which channel we can look. And you can see the spike here. Okay? And we plug this electrical signal to a speaker. So whenever you hear a click, it's because it's a neuron that it's firing. First of all, well, let's see if you can hear something. I don't know. If not, I can play back you later. Okay, doesn't matter. See? Okay, so when the bird is singing, you see there's this massive response, a lot of neurons, and whenever you have like this beep, 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 you hear the neurons like doing. So that is strong evidence that you have neural recordings that are matching also the rhythm while the bird is singing. And when the bird is not singing, then everything goes very quiet, okay? So we look at the data, LFPs while the bird is singing. So here is what the bird is singing, a representation in the sonogram. And you can see if the syllable is fast, you have these fast oscillations. When the syllable is slower, you have these slower oscillations. And the oscillations also, for example, this is a fast syllable, but not so fast as this one. And you can see that the oscillations are changing, okay? And again, if we plot the syllabic rate and the frequency of the LFPs, they are all well aligned in an identity curve, okay? So if you look at so this is just to show you that if you look at anesthetized birds or a singing birds, in all of them, you have that these rhythms in the brain match the rhythms of the behavior. So this is the main message that I wanted to that you take, that local field potential frequencies match the syllable repetition rates in auditory responses and during song production. Then we did some, because then the question is, what happened with the neurons? If you look at individual neurons and you do spike sortings, also these individual neurons are matching the rhythms. So whenever you see activity in the neurons, they correspond to rhythms in the brain. So there is a correspondence between single unit activity, single neurons activity, and these oscillations. Doesn't matter. This doesn't matter. So yeah, no. But this is so. This was the most important. So we have some time for question. I prefer that. And if then I will be here these 3 days, I can show you later. The main conclusions are that the importance of characterizing the behavior while studying motor control. The rhythms in the brains are synchronized with behavior, and these neural recordings suggest a motor gesture-related neural code. I wanted to thank all the people that is responsible for this work, my collaborators, Gabriel Mindlin and Margolias, my students, and some families. So thank you so much. Yes.
So, like the recording which is being made while the song is being, sung by the birds. So, is the recording due to this birds singing the song, or it could be the behavioral response? The recording which we are measuring, that could be a response of the bird which is singing and it's hearing itself.
Yeah, very interesting question. So the question is, are these recording the neural activity corresponds to the generation of the behavior, or are these recording just because they are hearing to themselves? So that actually depends a lot on the species of bird that you are recording. There are some species that has been shown that in this place where you're recording, because it depends on which place of the brain you are recording, that they kind of go ballistic. There are some species that they don't hear themselves. They just go and sing. Though these are like zebra finches. Canaries, on the other side, they can hear themselves where they are awake. So actually, we don't know if these are signals that are because they are hearing to themselves, or because if they are generating. When we look in detail, actually, what we are finding is there are some groups that you can have different, so they are all synchronized, but you can have different phases. So the phases of the synchronization, some they come before and some they come after. So our hypothesis is that you have in this region together neurons that can fire while they are hearing or while they are, because of they are producing. But the truth is, we really don't know. It can be both in this species, in this part of the brain. And I have to add that in these birds, they have in this region reported what they call mirror neurons. So neurons that they fire in the same way while they are generating the action, or while they are looking at the action or hearing at the action. And so it's also a thing of that these neurons are firing with the same pattern. Yeah.
So a deaf bird cannot learn new songs, right? Because
exactly. Yeah.
Will it be able to invent new songs by trying out different combinations of syllables? Because birds do that, right?
Yeah. So what they were so deaf birds don't. What happens with deaf birds is that they start degrading their song because they cannot hear themselves. They don't have this feedback, so they start like losing the ability. Not many experiments has been done with deaf birds. That's the other thing. So, for example, with humans, if you have an accident and you lose the ability of hearing, you can do some exercises in order to keep your capacity of, because in human also the speech starts degrading. So it could be interesting to do this. Can birds learn new or not degrade their song? The thing is that those are hard experiments and they hadn't been done in birds. So
because a deaf Beethoven composed 9 symphonies.
because of Beethoven. Yeah. Well, actually, yeah, because that's what we think, that he already has the music in their brains. Yeah, that could be the case, but we don't know. Well, actually, what we know, but that depends on the species, that there are some species that has a specific window in their lives, when they are juveniles, when they can have like these auditory memories that we call it. And that is close, and all what they have learned and that it's fixed in their memory, it's fixed, but they can get to these auditory memories throughout their lives. So something that they hear when they were juveniles and they start singing it like afterwards. So this can happen. But the truth is that with deaf birds, we haven't done much experiments.
So are there ethics issues in doing this kind of experiments? Can you define a bird and study?
Well, yeah, so yes, and that is changing. So the question is ethic issues. So you have to go through very strict protocols in order so that the university authorize you to do the experiments. In Buenos Aires, they are they follow the protocols in the United States, and they are very strict. And you have to guarantee that the bird is. To us, it's easy because we want the bird to be singing, so he has to be okay and good and in well capacity and happy in order to sing. If not the bird, if they are not right, they don't sing. That's a fact. But doesn't matter. You have to prove that everything is well done, and you have to justify a lot if you are going to deafen a bird. Why you are doing that? How many birds? So you have to reduce the amount of birds you use. And that's also changing through. It's not the same experiments you can do 20 years ago that the experiments that you can do now. Yeah, but yeah, that's all. Yeah.
I think you said you're correlating motor activity with the song, right?
Yes.
What is the motor activity? Does it cock its head?
Yeah, so motor activity, they have many motor activities and they are all correlated. So motor activity could be the respiratory activity. So the way they are, the pressure that you measure in the air sacs, so the respiratory activity, but you can also have motor activity so that could be, you can measure in the air sac pressure or in the muscles, so the respiratory muscles that are generating that behavior. So that is the motor activity that we are focusing, but also could be motor activity, the muscle activity in the vocal folds that control the vocal folds. That we are not studying in this experiment. So it's mostly like the motor activity related to the muscle activity that is generating the pressure patterns in order to sing. But they also do some movements, and with that also depends on the species. That is something we want to start doing in the next year. So recording to the bird and analyze those recording and see how we can
And well, not quite related question. There are many species that actually speak human sounds. There are birds that talk.
Yeah.
We hear the hill myna and others.
Yeah, parrots.
Parrots
Yeah.
speak. So are these, because to distinguish between whether it is responding to what it's hearing or what it's generating, that could be a way of differentiating in species that actually work.
Yeah, but all the birds that mimic the humans, those are learners. And one thing that is interesting, so for example, parrots, they can imitate humans because they have the appropriate tongue. So parrots have tongues as ours, so this tongue that can be, in general, birds has a teeth tongue. And that's why most of the birds cannot imitate the humans, maybe also because they are not interested in doing that. But yeah, so those are parrots. So parrots, for example, in Spanish, that we roll our R's with so they can do that because they have the appropriate tongue. But those are vocal learners and learners that can learn throughout their lives, so they can learn, And that's
you haven't done experiments with that?
Not ourselves. Those are very hard. There are some labs that do experiments with parrots, but those are very hard. So actually, they are more like they have names. So you have specific. So there was a very popular parrot that was called Alex. That he was like a super genius and could learn a lot and all that. And it was a disaster when he died because he eventually died. And so you have few parrots and you interact with them, but yeah, it's not easy to, yeah. Yeah, so we could do that. We didn't do it. So you can perturb the song. For example, you can damage a little bit the tracheal nerves, and they start acoustically. They will change the way they are vocalizing, but the rhythm would be the same. The problem is that whenever you perturb that, many things can be altered, and then it could be hard to interpret. But the truth is, we didn't do it. It could be one line of research to see, well, what's the difference and what are these neurons coding? And for example, this question of, are they responding? It's a motor act or it's an auditory feedback. You can disentangle with those kind of experiments. If they are firing at the same, well, probably it's not auditory experience. But then there's always, well, but it could be that it's just they're coding the rhythm, so they don't care about, well, I don't know. It's sophisticated. So it's not so easy when you perturb then to interpret the results, but you could do that. Okay. Yeah, well, that's it's a very nice thing. We discussed that a lot with Gabo, yeah, to say, well, do some some stimulation in this region to change the phase. One difficulty with this is that this is an extend region, so you have to tune how you're going to do these perturbations in order to have some, so it can be a sophisticated thing, but yeah, we've been discussing, so we would very much would like to do that. It's a sophisticated experiment to do, but we would like to do it, yeah. Yeah, but the problem is that when you do experiments in biology, you have to be very careful to control so what you're doing, and because then you have to interpret your results. So yeah.
So I wonder, within HPC, were the patterns different on different electrodes? Or was the rhythm sort of synchronous all over?
Yeah, so we always, so that's interesting. So actually what we find is we try to get to a specific region in HPC. And actually one thing that we find is that depending on the region, you could have auditory responses or not, for example. It for sure depends on different regions. That is in the experiments while the bird is singing. And we try to get to the same similar regions. When we were doing the anesthetized experiments, that there we can this kind of recording. So this where we can put an elite. So this was the bird is anesthetized, and we can do like different regions. And so each line here, it's a different stereotaxic coordinates, and we didn't find any differences. So in the different regions in HBC, and these were like long experiments, and we really do a huge grid, and we couldn't find differences. But this is an anesthetized bird. It's always, so this is just the brain responding to the auditory stimulus. It can be that during singing it's different, but in our hands they are always synchronized.
I think that the LFP
firing activity, or is it reflecting this input, synaptic input?
Well, that we don't know, right? So we don't know if it's local or if it's the auditory. It could be that the song is synchronizing this. So, for example, so what we are finding here is, so this, it looks like so these are the rasters of the individual neurons, and it looks like the peaks. So either if we do multi-unit activity, or if we look at individual neurons, or we look, so this, where the neurons are firing, is where you look at these peaks in the LFP. So if you look at it like this, it seems that LFP is reflecting the local activity, but it can also be that it all is well synchronized. It also could be that it's the inputs, and the neurons are responding to these inputs. So with LFPs, you don't really know for sure. Yeah. Yeah, okay. Super happy with all the questions. We will be here these 3 days, so let's continue the discussions. Thank you so much for your interest.
The next speaker is demanding to.
Yes, thank you so much.